innovate. And the post-graduation students submitted a pitch video to communicate their work to society. Before coming to a close with these remarks, I would like to remind that the closing session will be on Friday at noon. And I would like to ask our moderators to stick to our schedule and not to let any session overrun. I sincerely hope you will enjoy today and the next 2 days of debate and networking. Thank you all for your participation. At this stage, I would like to introduce Dr. Patrícia Sartorelli, Head of the Chemical Biology Postgraduation Program at Unifesp. Welcome, Dr. Patrícia.
Thank you, Lucia. Good morning, everybody. I will share my screen. I would like to welcome all participants to the 3rd Congress Chemical Biology, CCB, and 4th Symposium of Biological Science Department, version 2021. As Professor Lucia said, in this year, the theme of CCB is Chemical Biology for Maintenance of Human Health and Environment Equilibrium, and will cover a wide range of critically important sessions. In addition, I would also like to emphasize that the CCB will take place entirely virtually due to the pandemic of COVID-19, and this will allow interaction among scientists and students from different areas. Our focus is on integrating the chemistry perspective to biological questions that will bring up new insights to human and environmental problems that may provide new solutions accessible to all. Graduate program in chemical biology is part of one of South America's largest universities, Unifesp, and has a critical role to play in contributing to human health and ecological equilibrium. Just following the Congress activities occurs the Symposium of Biological Science Department. For this event, the organizing committee invited Brazilian speakers who will debate important aspects related to the feeling and teaching experience during pandemic times. Both events are offered in an integrated meeting for which will be expected about 200 Brazilian and foreign attendees, including research, graduate, undergraduate, and undergraduate students. The first 2 days will be lectures by guest researchers, and the last day will be dedicated to mini course. The complete schedule can be accessed on the event website. I would also like to talk a little about the graduate program in chemical biology that has a major goal providing top quality postgraduate training in a range of research areas that address knowledge in the biology and chemistry interface. Our program aims to integrate the knowledge of chemistry that apply to biological systems, including studies with molecules, cells, organisms, and natural processes responsible for the diversity of life forms. So, the chemical biology graduate program research areas include chemical biology of systems. This area of research involves the study of behavioral, chemical, and biological phenomena associated with functional and structural modification of the system. Chemical biology in cell models. This research area includes cellular and molecular studies that aim to evaluate key processes in neuroscience, endocrinology, oncology, microbiology, and physiology. Furthermore, these studies aims to evaluate the effects of bioactive substances and signaling molecules. And the third area is chemical biology of natural and synthetic products. This area aims to isolate, synthesize, identify, and characterize natural and synthetic products with biological activity that may have therapeutic application. We are currently 23 supervisors, and we have 53 regularly enrolled students, 28 of whom are the master and 25 in the doctorate. This program has been promised in terms of publication, fundraising, international projection, and training of students. To illustrate, in the last quadrennium, 2016 to 2020, 354 articles linked to the program were published in the index journals, being 112 in the stratum A1 of CAPS. Moreover, almost 2 million reais
Recording in progress.
Moreover, almost 2 million reais were collected in addition to $388,000 in projects coordinated by professors who supervise in the program. And more than 53 students were trained, being 33 master and 20 doctorate grades. Data from program graduates show the capacity to train qualified human resources in addition to competence of graduating in new groups, in teaching or research centers, where they are linked. It's also possible to identify actions to promote local and regional development through its performance as trainers in secondary and higher education, or even who have worked in their specific areas in industry or companies for development, innovation, and or innovation. The data also showed that 25% of PhD graduates are professors working directly in nucleation development, research, and as professor in the public or private universities. And finally, some works developed in the program were highlighted in the scientific community and the media for approaching research with an innovative character and application in society. For more information about the program and the research developed by our supervisors, you can visit the program web page. Finally, I would like to thank the organizing committee for the inestimable work done to make our event possible. And I wish you an excellent event. Thank you. Professor Lucian. Rafaela, can you unshare my screen?
Sorry. Sorry. Thank you, Patricia. Thank you very much. Now, I am glad to present our first speaker. I want to introduce Dr. Marcelo Lima. Is Dr. Marcelo? Yeah.
Yes, I'm here.
Hello, Marcelo.
I am.
Now I will introduce Dr. Lima. He is a biologist from Federal University of Rio Grande do Sul with a PhD in molecular biology from the Federal University of São Paulo. Dr. Lima has been working with host virus interactions and the related macromolecules with an emphasis on viruses, such as Zika and SARS-CoV-2. Some of his activities include lecture in the biomedical science at Keele University and biochemistry professor at Unifesp. I would like you all to participate with questions that can be written in the chat and will be presented to Dr. Lima at the end of the talk. Quem quiser fazer perguntas em português, tanto no YouTube quanto aqui, pode colocar no chat em português, que a gente vai traduzir e colocar para o Dr. Lima. Welcome, Dr. Marcelo Lima.
Okay, thanks a lot. Good afternoon, everybody. Actually, good morning. I'm just 4 hours ahead. So yeah, my head's already post-lunch. But thanks a lot for the invitation. Thanks, Lucia, for getting in touch a few months ago. And, I said right away that I would be, really happy to take part. I was part of the São Paulo campus in Unifesp, but, it's the same university. So it's always really nice to go back home somehow. Of course, because of the pandemic, all of these things need to happen remotely. But it's always nice to see some friendly faces and people that I used to work with some years ago. So as Lucia highlighted, I was a professor in biochemistry at the molecular biology division at Unifesp for about 4 years. And I moved to England in 2019. So that's where I'm based now as a lecturer in biomedical science at Keele University. And I'm also part of the Glycoscience Center for Research, which has been recently created here, that we have a group of 7 of us that are interested in glycoscience as a whole. Of course, like everyone with different capabilities and expertise. But we try to come together and try to tackle some of the scientific problems. And for today, I'm going to be talking about the work that we've been doing on COVID for the past essentially 18 months. And it just shows that even though none of us are virologists, we came together to find, different solutions for, the problem that everyone knows about it. And to be honest, everyone's probably sick of it already. It's good that vaccinations are going well and we're going back to some kind of normality. But we all know that I would probably say we're far from getting rid of it completely. I would say it's probably going to be around for quite a bit of time. And it just emphasizes that we need to keep doing research on it. And not only research in terms of the virus itself, because essentially if you break down these different viruses, they are different molecules. So everyone with interest in molecules in general can contribute quite a lot. I'm far from being a virologist. My background is molecular biology and especially on carbohydrates. But we do work on some of these models, and we try to understand and essentially give some kind of solutions using our background knowledge. So I'm gonna share my screen if that's okay. Is that okay, Lucia? And let me just find the button. Share screen. Okay. And now I need to find which one it is. So that's the one. Can everybody see my screen? Can you guys see my screen?
Yeah.
Okay, Marcel, yes.
Yeah, perfect. Okay. So as Lucia said, and as I mentioned, I'm part now of Keele University, although I still collaborate quite a lot with my old lab in São Paulo. And this is just a picture of the campus. So the first question that we normally get, where is Kiel? And essentially, this is a map of the UK. And essentially, Kiel is at the Midlands, is in the Midlands of the country. Just to give you some kind of perspective, it takes about 1.5 hours from London in a train. And we are roughly speaking about an hour away from Manchester, Liverpool. It's a very small village. So I live in a countryside, which is really nice. Those typical countryside houses, garden and all of that, because there's a lot of green around here. There's no big cities around. So it's pretty great. And in a way, I was really, in a way, happy to go back to like a countryside after 11 years in São Paulo, living in the concrete jungle that we all know quite well. And today I will talk about some of the work that we've been doing for the past 18 months that relates to heparin sulfate. And I just gave this kind of like slightly odd title, but just to say that there is a sweet side in coronavirus treatment, because these two molecules are carbohydrates, so essentially they are sugars. So it kind of goes a little bit away from what we hear on a daily basis in terms of vaccines or protein inhibitors or protease inhibitors, small molecules, and all of these things. And this is essentially the work that we've been doing for the past 18 months. But of course, to start, before we get into the actual coronavirus, I would like to introduce a bit of the topic, especially around carbohydrates, because essentially there is a problem when it comes to what we teach in undergraduate for essentially what we teach our students around these molecules. And a lot of the textbooks just mention them, briefly, and they just say that these molecules only exist to soak up water in joints and things like that, in tissues, which is far from being true, because they do a lot more, and I will show you some examples. Okay, so basically, we're gonna go back to the start, as we all know about the central dogma of life, where a DNA molecule can essentially replicate itself. Of course, it has information to produce all the components that it requires to replicate itself. But some of this DNA is going to essentially become RNA molecules. These RNA molecules will become proteins, and eventually these proteins will organize themselves and form the different cells that we have in any given tissue. So this kind of like gave birth to the term central dogma of life, in which everything that we need to know about a certain organism is contained within the DNA. But there's a little problem here, because there are a lot of questions around how the different proteins organize themselves into different cell types, and also how the different cell types organize themselves to form different organisms. So we can probably now guess that there must be something else apart from essentially the information that is directly encoded within the DNA that needs to come here, to come together here in order to have different organisms being formed. And I always like to use this example that we can't just rely on the genome itself, because it's not really possible to explain biology just using the genome. And this is just a very simple example where in a plant, we have, roughly speaking, about 26,000 genes. And whereas in, us or monkeys, we have, roughly speaking, about 30,000 genes. And in this case here, there's even a 99.9% identity, and we are very, very different. So if we just look at the number of genes, or essentially the information contained within the DNA, it's not really enough to explain, all of these differences. So it goes beyond that, and that's where I would like to make my first comment. I do understand, and I appreciate why DNA is so studied, and a lot of people focus on it. But there are many different molecules that need to be looked at apart from the DNA. Okay, and when we look at what happens in terms of the information that is encoded within the DNA, As we move away from, the DNA itself, we face a increasing functional diversity. And this functional diversity essentially is the result of increasing chemical diversity. So RNA and DNA and RNA, they're essentially they're very similar molecules. Chemically speaking, there's not much going on in terms of differences. But then when you get to proteins, you start getting close to hundreds of thousands of different chemical entities. And of course, this is going to have a lot more functions associated with them. And by the time we get the proteins, they're modified. So essentially the post-translation modifications, we are facing millions of molecules. And you can probably already guess that these molecules will have many, many different functions. And a lot of my talk is going to be around essentially some of these molecules that change the structure of these proteins. And essentially, I will talk about carbohydrates that change the structure of proteins via a process called glycosylation. So essentially, the addition of carbohydrate units into a protein backbone. Okay, but again, just highlighting that we are here, when we get here, we're facing essentially millions of different chemical entities that will have many, many different functions. So if we think about what the real information flux is when it comes of, different molecules coming together, forming different cells and forming different organisms, that's probably slightly more realistic. Yes, we do know that DNA can lead to RNA to protein. But when it comes to here, essentially all of these different components at some point will interact to form the different components of a given cell that eventually will come together to form a different kind of organisms. So remember what I mentioned a few slides ago. Yes, by all means, this needs to be looked at, but we simply can't ignore all of these different molecules that do exist in any biological system. So that's why now, more than ever, we appreciate that only sequencing the DNA, as we already done anyway, like 20 years ago, is not really enough to understand all there is to know about a specific organism. That there is a lot more behind this. And one of the challenges in doing these kind of studies, especially around carbohydrates, the one that I'm going to focus on as we move along with my presentation, is that there's no real way that we know what these carbohydrates are going to be based on the information written on the DNA. So chemically speaking, to analyze, to understand what these molecules are, it's much more complicated because it involves very laborious techniques, things that take a very long time. And sometimes there are things that we can't even do nowadays because essentially there's no technology for it. Okay. And going back to, the main thing that I'll focus from now on, I'm just going to use the example of one class of those molecules that kind of like form the information flux. And these molecules are called the glycoconjugates. So essentially, the name says it all. So it's some kind of molecule that has a glycan component, so essentially like a sugar component to them. So this is just a cartoon of what a cell membrane kind of looks like. And as we can see, here's the cell membrane. There'll be loads of different molecules associated to our cell surface. But the common feature of all of these is that they have carbohydrate units attached to them. So we can probably start thinking that our cell surface is covered with carbohydrates, hence my title, The Sweet Side of, in this case, the coronavirus treatment. So basically, all of these molecules will have different monosaccharides forming different polysaccharides as their main chemical composition. And this is very variable. It will change from tissue X to tissue Y, and a lot of the reasons why the profile changes are still unknown. But this does exist. And someone could ask, oh, okay, but this is only a cartoon. Does it really look like this? And essentially, when we look at a cell, so this is just a normal endothelium cell being looked at confocal microscope. So we use different staining methods here to label different components of the cell. So typically speaking, the usual blue DAPI for the DNA nuclei. In green here is just the actin filaments. Then in red here we have a labeling using antibodies specifically targeting those glycoconjugates on the cell surface. And as you can see here, this is a 3D projection, and you can kind of tell that those glycoconjugates, they do kind of form, they do cover the cell. We can even see the shape. So actually that cartoon that I showed, it's a good representation of what happens in real life. This is just another example. These are muscle cells, so different cell type, but kind of like the same feature. And if we flip, this projection 180 degrees, essentially, so we're looking at the side, we can see that in green here, covering all of the cells. These are essentially carbohydrates that are being stained with using a different antibody. So even a different kind of labeling that we used from the previous slide. But again, something that it's been designed to look at carbohydrates shows more or less the same profile. So the cells are covered with carbohydrates as a whole. So going back to my cartoon, it just shows that indeed, that cartoon is a pretty close representation to what happens in real life. And ultimately, just to show the finer example of where these things are, and again, to highlight the fact that that cartoon is actually true, this is just staining for a structure that we call a glycocalyx. So essentially, all of the carbohydrate components that decorate the cell surface of many different cell types and tissues in a human body. And it's just showing here that we have a quite thick layer of glycoconjugates at the surface. So these are essentially blood vessels. And as you can see here, we have the endothelium cell layer here. And all of that are those glycoconjugates, those different ones that I showed before. And here at the bottom is just an isolated lymphocyte, so essentially an isolated cell, but it has the same kind of feature. At the top of the cell membrane, we do have that thick layer of glycans covering. So you can kind of wonder where I'm going with this. So essentially, if something's floating around in a blood vessel or in any tissue, if it needs to go into these cells at some point, it's going to have to bind to some of these carbohydrates. It doesn't sound like a crazy thing to expect, given the fact that they are indeed covered in these different cells are indeed covered in carbohydrates. And going back to the problem, essentially, a lot of research goes around determining the structure of these carbohydrates to associate with a specific function. And to determine that kind of correlation, it would be very nice to have, chemically defined carbohydrates that we could test. But unfortunately, unlike DNA and protein, that we know what the code is, and we can essentially nowadays synthesize anything that we want in a lab. The process that dictates how these carbohydrate chains are synthesized happens, at the Golgi interface, and we simply don't really understand how this process works. So there's no such thing as doing recombinant glycans or recombinant glycoconjugates. We can do a couple of things using semi-synthesis or synthesis or chemoenzymatic enzymatic synthesis, but we are really, really far away from actually accessing all of the structural combinations that exist in a organism. Okay. And to make our life even more complicated, these molecules, as they're synthesized, they are quite diverse. And this is just in a slide covering heparin and heparin sulfate, how diverse their structure can be. And this is just an example. We can break down this structural diversity into the composition aspect. So we essentially going to have a repeating disaccharide building block or backbone, if you like, of these two monosaccharides. But if we break it down, there's always going to be a uronic acid, but the uronic acid can either be the D-glucuronic acid or L-iduronic acid. So that's already two possibilities. When we look at the glucosamine right next to it, so there's going to be a glycosidic linkage here. This can be an acetylglucosamine and sulfoglucosamine or just N-glucosamine. So again, you know, 3 more combinations. And there might be sulfates added in these 4 positions. So you can kind of start like making some maths around how many possibilities we have for a given disaccharide. The thing is, as we start looking at the polymer as a whole, there might be you know, some sequence or block organization taking place within the chain. So you might have a certain chain that will have different blocks of different structures. This can happen, and it's known to happen in some cell surface heparin sulfates, but also the glycosidic linkage, you know, the linkage that essentially links one monosaccharide to another, they can have torsion angles. So essentially can have, you know, torsions going this way or going that way. So it just adds slightly more to the problem of analyzing these molecules. When it comes to the uronic acid, specifically around the iduronic acid, these monosaccharides, they can change their space orientation. So essentially going back to very old organic chemistry, you know, the cherry and boat conformation. So you can have things going from one conformation to another in solution. And this happens quite a lot. And I'll actually show you guys an example of this taking place when the interaction happens of a heparin sulfate analog with the spike protein of the SARS-CoV-2. And when we put all together, essentially, we kind of have, you know, these cartoon representations of what the molecule looks like. But again, if we add all this up, this can be very, very complicated in terms of what it actually is, chemically speaking, conformationally speaking. And all of that, if we combine, we have essentially 48 possible combinations for a single disaccharide. So very, very complex, you know, way more complex than for instance, the possibilities that we have for DNA or RNA. And when we look at this structure diversity, all of this diversity essentially leads to many different biological functions. And this is just a network analysis of the different biological processes that these molecules are associated with. As darker as it gets, there's more essentially relevance, and the larger the circle, it just means that there are many more proteins that actually bind to these molecules at some point. And as you can see, these molecules are involved with basically anything, processes that go from, you know, cell signaling, cell communication, to essentially system development, organ development, even how you move, behavioral and wound healing and all these things. And this is a result of their vast chemical diversity. You know, there's a direct correlation between the amount of chemical information that these molecules can encode, you know, throughout them, and also the many different proteins and that will be associated with a given biological process. And every time you get a different structure, you might have different binding, therefore you're going to have different activity. So it's quite complex, but it's really fascinating to study these molecules because you can essentially go into any field you like. And going back to the glycoconjugates, because as I already kind of gave you a hint, if we have something floating around, potentially, you know, whatever it may be, as a protein or a viral particle or a bacteria or anything, at some point it's going to probably bind to some of these molecules that do exist in a cell surface. So this is just a pretty small summary of different biological processes that these molecules are known to play a role. And as you can see, they do play a role, for instance, in uptaking things from the outside to the inside. Of course, in this case, it's just an example of a lipoprotein, but you can probably guess where I'm going from, where I'm getting to from now on with the context of coronaviruses. Okay. And, jumping into the actual main topic for the presentation, once we look at the human coronavirus structure, we've seen like many different cartoons, and we probably know on top of our head what it looks like. But basically, a lot of people have been studying like crazy for the past 18 months. This protein is called the spike. And essentially, that's what gives the name for these viruses anyway. Because the spike protein is the one that it seems to play the biggest role in infectivity. So basically, the ones that are going to interact with different cell receptors, and essentially we allow the coronavirus into the cell, and then once it's in the cell, it hijacks all of our molecular machinery just to make more viral particles. And as we know, and has been highly published and talked about, everyone became a bit of a specialist in how this happens. And it's good that people know things, but it also became a bit dangerous because everyone had an opinion about it, even though they had no background knowledge on it. But oh well, as we all know, one of the main receptors, or the most famous receptors for the spike protein, which eventually means for SARS-CoV-2, is ACE2. Okay, so once it binds to the cells, it goes into the cells and causes the infection. Therefore, it causes the disease progression. As we all know, a lot of people don't really have much problems with it, but it caused the problem that we all know about it. So it's not, by no means, like it was said last year, by no means is just a flu, because it really isn't just a flu. And as any good virus, it evolved, and essentially, there's different mutants. They acquire different characteristics, and a lot of these mutations happen at the spike protein, meaning that, for instance, they had increased contagious window, or increased virus shedding, or increased environmental stability, and this is where most of the research that I've been doing for the past 18 months takes place, increased binding to the host receptor. And of course, a lot of it is around ACE2, but you can probably tell that I'm not only going to be talking about ACE2, because there are different receptors. Okay, just a little background. I'm sure we all know this quite well. Just to highlight the how dangerous some of these mutants can be. So, for instance, the reproductive number for the wild type, the first strain that was isolated in China, is about 1.1. When we look at the B117 variant, it goes up to 1.74. And now there's even what they're calling the super Delta variant, or something along those lines. I kind of lost track how they named them. There's even more infectious, and it's around the mutations on the spike protein that affect how it binds to cell receptors and many different things. And the funny thing is that this is not really a new problem, in the sense that we've had issues with different coronavirus back in the day, and of course everyone knows the many different problems that we had with the flu. But just as a comparison, we had the SARS 2011, 2012, something like that, or even before that. And we also have the MERS, but these two were really isolated to specific regions. of course, this goes back to, 10, 15 years ago. We weren't as mobile as we are nowadays in terms of like we didn't travel as much as we do nowadays. So who knows if this would have happened in a globalized world that we have nowadays? Who knows where what would have happened? But what we've been dealing with for the past 18 months is the SARS-CoV-2. And, these numbers are just rough estimates. Of course, it depends. But one of the things that we can say, at least, essentially, is that we still don't really know where we're going, and we will have to wait and see what happens in the next maybe 6 or 12 months to kind of come up with numbers. But basically, this is the beast that we're dealing with now. And just looking at the spike protein more specifically, is divided into subunits, and within the S1, so essentially the subunit 1 is the bit that we call the receptor binding domain for ACE2. So essentially, is within the protein, there's a specific region of that domain that looks at the ACE2 as a cell receptor. And essentially, we were quite anxious when the whole thing was described. And I remember January last year, we we're kind of waiting and anxious for the sequence to be published. Once it was published, we started to look at what the sequence was and what the protein was going to look like. But before going into that, it's just, this slide is just to highlight the many different mutations that have happened, and this is ongoing anyway. But yeah, so when the sequence was published, and of course, like we all know about the ACE2 as a cell receptor and the protease activation that the spike protein happens, the protease activation that takes place for the spike protein essentially kind of like it's processed, and then it binds more to ACE2 and whatnot. But we also knew that these kind of molecules that we study, essentially the heparin sulfate, they do act as viral cell receptors. And when the sequence was published, we went ahead and started doing a lot of sequence analysis on the protein, and specifically on the region that we found more potential hits of, essentially more domains that we know that bind heparin sulfate. So we used our knowledge in essentially studying different heparin binding proteins. For instance, FGF1 it's a very known model. And we use the same techniques, the same bioinformatics techniques, to identify potential binding sites for heparin sulfate on the surface of the spike protein. And of course, when the sequence was published back in January, if I recall it correctly, we started the process of essentially all the cloning to express the protein. So we were, I think in the UK, we were one of the first groups to express the spike protein recombinantly in a lab. So essentially, we donated reagents, and especially these proteins, to several different groups here. And also, I shared plasmids and things also with the group in São Paulo as well. So I sent a lot of reagents in terms of plasmids, vectors for protein expression over there. And we identified many regions that would potentially bind heparin sulfate. So these are all in blue. And even one of the mutations that happened from the bat into the SARS-CoV-2 that we have, it could be highlighted as a GAM function, because the mutation was to gain an arginine at this position, and arginine is one of the amino acids that are involved with the binding with heparin sulfate. So we've done that. Of course, that this is all bioinformatics, and we needed to prove that. So with the recombinant protein hens, we did all the studies that we could do at the time, and a lot of it was around SPR. So essentially, we attached the the protein to a surface, and we flow the ligand, and we test the binding, and we saw that it actually bound, and the binding was very, very strong. We're using different structures to compete the binding off, and we kind of determined some kind of a correlation between a structure and activity, but it wasn't very clear because they seemed pretty flexible. So many different sequences, going back to the structural diversity that I mentioned at the start. Many of those sequences did bind, and we had, roughly speaking, the same results. And we also did a lot of circular dichroism just to see conformational changes in the protein when it was bound to the carbohydrate, and we could see a lot of changes on the CD profile. Meaning that the protein conformation is changing when it binds to heparin sulfate analogs. So it just kind of highlights that actually, yes, these molecules are indeed a cell receptor. So this was proposed back in, I think, like late February, early March last year as a preprint. And many different groups got involved in this. Eventually, we published more around this, but different groups also essentially showed what we first showed, and saying that yes, heparin sulfates are indeed SARS-CoV-2 cell receptors. And we moved on to more specific experiments, trying to understand the binding. Again, this goes a lot with my background in essentially molecular and structural biology, and we did a lot of NMR and molecular modeling, and we determined the binding sites on a protein. So essentially, you narrow down the amino acids that interact, and a lot of them go back to that first figure that I showed. Those blue areas on the surface of the protein that do interact with carbohydrates. And also, we wanted to see what happens in the carbohydrates, because it's not only the protein that changes its shape once the carbohydrate binds to one of these targets, one of the proteins. It also changes. So this is just an experiment where we determine where the protein is binding to carbohydrate. So it's just a little a scheme for how the experiment happens. So it's done using NMR, and we magnetize the protein, and then the protein transfers energy into the carbohydrates or the ligand. And as the closer the carbohydrate is to the protein, the stronger the magnetization transfer. Essentially, the stronger the signal. So we can determine, for instance, using a carbohydrate chain where the binding is taking place, and it kind of agrees with what we already knew from the SPR, that the binding is quite flexible. Essentially, it just likes to bind, and it really binds to the entire chain. There's no real direct structure to function correlation in a sense of a specific structure or a specific sulfate or a specific monosaccharide is going to be responsible for the entire binding. It's more to do with potentially the conformation and how the molecules behave in solution than a specific saccharide. And this is essentially what we can see. So we have signal that associates to the different monosaccharides that we have in oligosaccharide that we used to test the binding. And going back to the change also in conformation of the carbohydrate, remember that I mentioned that dehydrogenic acids, they can change from the chair into the boat conformation and whatnot. And this is just an experiment that we can test that. So we can kind of determine the distance between the different protons around the monosaccharide ring using an experiment where essentially there is energy transferred through space. So we just measure the NOEs, the nuclear overhaul effect on a molecule. And we can see that this is just the oligosaccharide on its own. When we do the 1D selective nosy experiment, essentially I'm looking at these two positions, so position 5 and 2 and position 5 and 4. So I can calculate the energy transfer that gives rise to the NOE signal. And when it's essentially free, not bound, there seems to be more of this conformation. But when the spike protein is added to the solution, so essentially when the binding takes place, we have an increase of this conformation. And essentially, we can tell this because the NOE signal from I5 to I2, so essentially these two here, increases. So it means that these two protons are closer. That's why we have more of that signal, meaning that there is a conformation change also on a carbohydrate. So again, just highlights the fact that, yes, we need to look into these things in detail as well. But of course, like, the main question is, okay, all of these molecular interactions taking place and all of this, does it really matter? Does it really make a difference when it comes to, the viral infection and whatnot? So we did 2 experiments, and now we've done many others. Even now we have animal models data, and we are pretty confident that it actually works. And I'll show some examples also, some clinical trials that took place. But when we use heparin and different heparin sulfate analogs, we can inhibit the infection in cells. And as I said, we have now data on animal models. So these are different concentrations of heparin, and we were able to inhibit the infection quite a lot. And we did the experiments alongside using the SARS-CoV, so the one that caused the first pandemic. No, it wasn't a pandemic at that point, really. It was isolated. And we do have inhibition, not as as nice as this one, but this seems to be a common underlying feature, and which is the spike protein binding. Although the spike protein from SARS-CoV is very different than SARS-CoV-2. And we also did experiments where we tested the binding of the RBD into cells, and essentially we could potentially say that actually it's via this mechanism. So essentially, you were able to inhibit the RBD binding to cells by treating the cells with heparin as well. So we're just detecting, how much of the RBD would be bound to the cell surface before and after treatment with heparin. Okay, and the interesting thing about this whole story is the fact that alongside, of course, like the infection itself, a lot of the problems and a lot of the issues that the people have with SARS-CoV-2 infection, essentially with COVID, is that it creates a lot of problems associated with thrombosis. So a lot of these patients have thrombosis, and very, very bad ones. And essentially, that's what, in many cases, kill them. And we all know that heparin is an anticoagulant. So actually, we're dealing with a drug that has the antiviral activity, but also has the anticoagulant activity. So it's got 2 activities in one molecule. And this goes back to the comment that I made at the start, that because of these molecules are so structurally diverse, they do many different things in the human body, or on essentially any organism really that do have these molecules. It's just to highlight that a lot of the issues around the COVID was around problems related to blood coagulation. So we can actually have a pretty good drug, not to cure it, I know that, but at least to make the whole process slightly less complicated. And we have to put this into perspective. Back then, we didn't have vaccines as we do nowadays. And moving forward, as these new variants show up, we need to have treatments. We need to have ways to treat people until we can develop and test a vaccine against a specific variant that, for some reason, is able to escape immunity. So essentially, as I said, a lot of it's not only the antiviral activity, but also the fact that the heparin has this capability of treating the intravascular coagulation that happens in COVID patients. There's even a lot of trials going on. I am involved in a few. They're trying to deliver heparin as a nasal spray. So essentially, it will go straight into our lungs and try to inhibit the infection as it may be happening. So as a very fast way to inhibit. So this is ongoing at the moment. And recently, there's been 2 big clinical trials that were published to do with treating patients with COVID with heparin. And all of them show that actually it works. It prevents deaths quite well. It doesn't work as well in critically ill patients, probably because it's already too late to try to have an intervention. But when you do treat patients when they're not at that stage of going to ICU beds and things, it gets better. And just to show how important this whole thing is, now, if you look at the WHO, heparin is one of the drugs that is listed as a treatment for COVID-19. So these are the 2 trials that were published recently that show that it does have an effect. And now we can because we have access to these samples, we can look into viral load, what happens into protein networks, and all of these things. So there's a lot to investigate. And essentially, I'm looking into as well, the binding of the different variants into heparin sulfate. So we have in our lab here a lot of the different spike variants expressed. They were looking into the structural biology aspect of it. I still collaborate, as I mentioned a few times, with the colleagues in São Paulo, and we're trying to do the NMR backbone determination using NMR down in Brazil, but the protein is quite difficult to express. So there's a lot of things happening as we speak in this front. But just to show you, of course, I didn't do this myself, and especially the work around COVID. It was done in collaboration, a lot of collaboration actually, with different colleagues at here at Kiel, but also the group in São Paulo and also the group in Liverpool, in which I've been to several times, and some parts of my postdoc and PhD were actually done over there. But these are basically different places that I've collaborated with. Some of them I've been to. So I've been, of course, like here and here. I've been to Liverpool as well. I've been to Diamond. I've been to the States in Loyola, and I've been to the University of Florida as well. And I also spent some time at the Rosoni St. Milan. So it just shows that, I've been kind of all over the place. And of course, we can't do any of that without funding. And I'm very grateful. Most recently, of course, the UK funding agencies. A lot of it's been funded by the actual university, but we did have grants from the Royal Society and also the UKRI to do all of that. And of course, most of my life I was in Brazil, so I did have a lot of funding coming from FAPESP, CAPES, FINEP, and SIMPIC. And a lot of my research as well, especially 5-6 years ago, longer than that actually, it was also financed by the state, so the National Science Foundation and the National High Field Laboratory in Florida. And yes, and thank you very much. And I'm happy to take any questions, any comments. And I know that I am Brazilian, so I do speak Portuguese. So if you're not comfortable in asking a question in English, at least it's to me it's perfectly fine. I can respond, but I'll see what I'm told to do, basically. Thanks a lot. I'll stop sharing my screen so I can see myself again and see what's going on. And yeah, happy to take any questions.
Thank you, Marcelo. Are you hearing me?
Yeah. Fine.
Okay, thank you very much. It was a great presentation. And we have already a question for you. I will read it. Is there any evidence of the spike being recognized by the human rage since there are carbohydrates linked to it, could we think, do I read once? There are two questions.
Yeah, it's fine. I'll respond at the end. It's okay.
Okay, could we think about any other molecular entrance to the virus beside ACE2, mainly when regarding the carbohydrates you mentioned as present into SARS-CoV-2 proteins?
Yes. So I'm going to be very honest. I don't really know what RAGE is, but I'm assuming it's a glycoprotein. I myself never looked into it, but it would make sense. It could be investigated, I would say. And in terms of different cell receptors now, yes, there are many other different cell receptors. So ACE2 was the one that was heavily studied to start with because of SARS-CoV, because it was already known that ACE2 was the main receptor. But now I think I could be wrong, but I've heard of at least like 4 or 5 different cell receptors for SARS-CoV-2. And it's interesting because there's even data that shows that there is infection independent of ACE2. So it just shows that indeed there are different entrance mechanisms, if you like, that don't even need ACE2 to take place. I can, Lucia, if you want, I can send you the links for all of these papers, and then you can share it with everybody. Because I know of at least like 4 or 5 different papers. Yeah, that are now known to play a role in the infection.
I think Professor Daniela Rando that had the questions, and she said that RAGE, RAGE, is a receptor for high advanced glycation end products.
Okay, yeah. I mean, I apologize for my ignorance. I never really looked into it. But yeah, I can even say I'll have a look into it and see if there is something around this. And I will reply to Lucia alongside the references for the papers. But yeah, it could be something worth having a look.
Do I read for you?
I can read it. It's fine. So someone is asking about TLR4. The same. I don't know specifically about this one. As far as I remember, it's not the ones that I've seen. But yes, it could be. It could well be that these may have some kind of involvement in a process, and we have to appreciate as well that even though we've done a lot of work already, if you think about it, 18 months is not a lot of time, scientifically speaking. So I wouldn't be surprised if a lot of these questions are answered within the next maybe 12 to 18 months, because there is a lot of research going on, and it might well be that I don't know, in 6 months' time, someone comes and says, oh, yes, the toll-like receptors are indeed another cell receptor for SARS-CoV-2, because it is still ongoing. I know that we want to go back to normality, and I'm really, really happy the vaccination has been great, because we can function. I haven't seen my family in Brazil for 2 years because, we weren't allowed to travel. Now recently the borders are open, so I'll probably be able to see my family in January, but we're not over with it yet. There's a lot still to happen. I think I just hope the vaccines keep working so we can kind of function, but we can never drop our guards 100%. Yeah.
And since you are deep in this kind of studies, Professor Monica Teles asked, in your opinion, what is the biggest challenge to deal with the SARS-CoV-2?
I would say now is the lack of knowledge around how these escape variants will behave, essentially. because I remember a lot of people when the whole problem started saying, oh, this SARS-CoV-2 doesn't seem to mutate a whole lot. I don't know where they got that information from, because we simply didn't know. And this is just my personal opinion. I was always a bit, cautious about making these kind of comments. And we've seen huge numbers of variants showing up for the past 6 to 8 months. Both Brazil and UK massively contributed to the problem, because the first VOC was described here as the UK variant, the Kent variant, and then Brazil had the issues with the P1 and also others. So the challenge really is to see how long the immunity lasts when it comes to vaccine, which leads to the second biggest challenge, which is developing a treatment that can be easily accessed, perhaps taking it home, that could prevent a lot of people going into hospital, because we just know that we don't have the capacity. And it's not a problem of Brazil or the UK or America, anywhere. Like once the infections, once the infection rate gets high, we just don't have enough beds to take everyone that gets ill. And that's why there's a lot of push now to develop, essentially pills that can be taken. And by no means I'm making any kind of correlation with the famous kids COVID, because we all know that was completely nonsense. I'm just saying that now Pfizer announced they're doing a phase 3 clinical trial with one of the drugs they identified, and there's a lot more going on. And I think that's going to be the challenge, like how long the immunity lasts, and also developing something that can essentially not stop completely, but something that can make our life easier, and essentially means that we'll be able to live with it, or to cope with it, essentially.
Yeah. Professor Nilana asks, what is the isoelectric point of the spike molecules that you have been tested?
That's a very good question. I did calculate it at some point, but I completely forgot the actual value. I will have to look into it, and then I'll reply to you, Lucia, because I forgot the actual.
You can send me all this, and I will.
Okay. Yeah, no, that's great. I apologize, but I did look into it. But yeah, it was something I tested once, and that was it. I don't remember. I'll have to go back and look at the value.
There is a question about applying an intermediary.
Yeah. So this is actually the $1 million question that a lot of people are trying to answer. And the second clinical trial that I showed essentially proved that a higher dose, a therapeutic dose, is more effective in treating the conditions. And when it comes to low molecular heparin and enoxaparin, it's the same. People are testing it. When it comes to the benefit, of course, like looking at the risk and benefit, we do have to appreciate that these molecules can lead to bleeding. So it goes back to the fact that a lot of these things need to happen in the hospital settings. It can't be taken at home. Because I know at some point it became quite popular. Oh, let's just take heparin. And it's not as simple as that, because it is a drug that, some people will eventually develop a bleeding as a response. So as long as it's done in a hospital setting, and then of course, if you do have the prescription to take low molecular heparin at home, there needs to be looked at. And again, I'm not a physician, so I'm not in position to say do X, Y, or Z. I think it's going to be a case by case situation, but there is the risk of bleeding.
Okay.
Should I go to the next one? Someone talking about the diversity. Yeah, so you mentioned the chemical diversity of carbohydrates with respect to the type of bond. In the case of the inhibitory effect of heparin against coronavirus infection, do you believe it is essential that the bond between monosaccharides is beta? Okay, so the type of glycosidic linkage. So essentially, the beta glycosidic linkage, beta 1,4 glycosidic linkage within heparin, it's one of the bonds that exists because you can also have alpha 1,4 depending on what monosaccharides you're talking about. From all of the sequence that we tried so far, that doesn't seem to this seems to be pretty promiscuous. But again, we haven't really tested very defined sequences, simply because a lot of this was done using what the drug is, which is complex, and it has basically everything. But your question is quite interesting because we are now, and we actually have some of these molecules, and we've been studying recently, not for the spike interaction, for other things. Instead of the typical O-glycosidic linkages, we have chemically synthetic heparin analogs that have an S, so a sulfur. So it's a S glycoside, so like a thiol glycoside. The reason why we're developing this, because it's quite unique in terms of how it behaves in solution, but also the fact that it's not degraded by our enzymes, so it becomes more stable. So it's like it would be like a chemically enhanced molecule in the sense that it wouldn't be degraded easily. So these things need to be looked at. But there's no direct structure function correlation. The only thing that we could find that was more defined was the molecular weight seems to be more important than the actual monosaccharide composition. And longer chains had better activity.
Okay. Marcelo, I am from the DNA side.
So apologies for all my comments then.
No, but it's great. It was great. And you really opened my mind actually to entry. My entrance test for Unifest was, I had a presentation on heparin sulfates. Mm-hmm. But one thing that struck me in your presentation was that we have 48 different types of heparin. So my question is, everybody has all these types of heparins, or it's this synthetic or physiological?
Yeah. So when we isolate from a tissue, and let me just draw a line between heparin and heparin sulfate, because heparin sulfate is the one that we have in the cell surface. Heparin is within our mast cells. But when I say heparin, I talk about pharmaceutical heparin, which actually extracted from pigs or cows. But in theory, we can find all of those 48 combinations within the mixture of the whole carbohydrate that we extract. So we do find all of those combinations in vivo. And don't forget about the torsion, the glycosidic linkage torsion. We know that they do change, but we can't measure them. There is a way to measure it using the Karplus equation. So we adapt it from proteins to carbohydrates, but it's not 100%. So actually, a lot of the research that I do, and especially around those S-link S-glycosides, we're trying to measure the torsion angles. There's not something easy to be done.
But
yeah,
so different people have different combinations.
It's relatively conserved under normal circumstances, but the structure does varies quite a lot when you go into different pathological conditions. So, for instance, there's a great change in the structure of heparin sulfate, for instance, in cancer. Different types of cancers. Of course, like every tissue is going to have its own main characteristic heparin sulfate, but it will change because of pathological conditions, age as well, and all of these things. So it's a quite dynamic molecule. And just to give an example, it changes throughout the cell cycle.
Okay.
We can't narrow it down exactly to what it is because every time we extract it is a mixture. But we know that it changes through the cell cycle. So the heparin sulfate that you get, for instance, as in S1, whatever, it's going to be very different than the one that exists in at the mitosis. So it changes very, very rapidly. Just as an example, we do know that the structure of heparin sulfate can change within 15 minutes.
Okay, but do you have population differences? Like, because I'm thinking about single
That's a very
nucleotide
good question.
polymorphisms
Yeah,
that could lead to
Yeah.
population differences, and this could be related to the different progression, COVID-19 progression in different people.
That's a
Because
very
you
good
have
question.
young people that die, and you have someone 100 years old that is alive. I know there are a lot of, how to say, components, but I wonder if this could be something.
This is a very good question. I have no idea, but I would say that no one ever looked at that. If there is population variation, if it's related to, I know the age changes, but for instance, if it's like race, age, where you are in the world, or these kind of things, I don't think anyone ever done this. And a lot of it's because it's so difficult to analyze these molecules that it's just something that it's not as easy. And don't get me wrong, it is a lot easier to sequence DNA.
Yeah.
Nowadays,
I know.
in 2021, we can't sequence a heparin sulfate chain. We cannot sequence a chain. So that's probably why this data doesn't exist.
And as I understood, you don't know how an enzyme defines the sequence of monosaccharides that will be linked.
We don't
know. We don't have this knowledge yet.
Yeah, no. So it's a process that happens at the interface between the ER and the Golgi. It involves about 30-something enzymes or more.
How many?
More than 30 enzymes. And we simply don't know what regulates how it happens. So there were a lot of studies, and there are a lot of studies relating, for instance, just gene expression. So up or down regulation of gene X, Y, and Z related to some kind of like a structural feature. But we also do work on the biosynthesis. So we went a step behind and said, okay, let's look at the Golgi. And the gene expression profile doesn't seem to be as important as the shuffling of these enzymes throughout the Golgi. So there's a quite complex process of enzyme recycling within the Golgi network that determines the structure that we get in the end instead of the gene expression. And this goes back to the comment that I made you. We have differences in structure within only 15, 20 minutes, which is impossible to have, the entire gene regulation cascade taking place to have more or less protein. So it needs to happen like that. So that's why the shuffling of the enzymes throughout the Golgi seem to play a role, but we don't know. We don't know what regulates. So yeah, this is actually one of the projects that we have as well, and a lot of it's being done in collaboration with the group in São Paulo still. Yeah. So yeah, it's as I joke around, it's nice because I think I'm pretty safe for having a job in looking at these molecules because we don't know anything about it. But at the same time, it's really challenging because a lot of things we simply can't do it because we don't have the technology to look at it.
Yeah. Well, I hope in the future you can, you can
we can sequence
a carbohydrate like you sequence DNA.
Yeah, or like having a similar thing of a PCR machine, just to produce.
Yes.
It would be amazing. It would be great.
At the bank.
Yeah.
And is heparin used for treating other coronavirus disease such as MERS?
It was never tested with MERS. There was data on SARS-CoV that inhibited the infection. But again, it never went to the clinical phase because the problem wasn't as big as it is now.
Mm-hmm.
Although it's quite common to have heparin in these kind of conditions, because essentially all of these patients do present a lot of coagulation issues. So it's common to have heparin at some point of the treatment. And not only because of the disease itself, but we can appreciate that some of these people are stuck to a bed for weeks and weeks. And at some point, they're going to end up having anticoagulants given to them because they are, stuck to a bed. But it never went to clinical phase when it comes to SARS or MERS, simply because, the problem was very isolated, and especially MERS, you only stay within the Middle East. So us from the Western world, we didn't really care about it because it never really affected us. It's sad to say, but it's true. With this, it hit everyone. And that's why we did what we did.
Yes. Well, I think there are no more questions. And I want to thank you, Marcelo, very much for the wonderful talk, for opening our minds to
that's great. Yeah,
of carbohydrates. It's wonderful.
Yeah, please don't say that they only exist to soak up water in joints, because it's, it just makes me sad.
Yes. That was my presentation. Joints. And I thank you very much. And I want to say to the public that the people that are watching us at the YouTube should make a registration at the PROAC site so we can access people that are watching our Congress. But thank you, Marcelo, and we will have a coffee break, and we begin again at 11 o'clock.
Okay, thanks a lot. And I will email you the references with the different receptors. And the other question, I can't remember. Let me just double check. The isoelectric point for the
yes.
Yeah, I'll get back to you with that. Okay, thanks a lot.
Thanks.
And
Bye.
Have a good day. Speak to you soon. Bye-bye.
Bye, Marcelo. Thank you.